This is a summary of a lecture I gave on apoptosis and the contents taken from the Hancock textbook. I guess the first thing to say about apoptosis is that it's a genetically programmed series of events. So apoptosis is a systematic process, and many of the characteristics of cells undergoing apoptosis have been well studied. So the question is, what are the factors that trigger apoptosis? So effectively, it's a balance between positive signals which encourage cell growth, such as hormones and growth factors, and negative signals which trigger apoptosis, such as cell damage by ultraviolet radiation, oxidants, and internal signals as part of development, which also trigger apoptosis. In contrast to apoptosis, we can have cell death by injury, such as mechanical damage or exposure to toxic chemicals. Now, when cells die by this method, they swell and they leach their contents out into the organism, which causes inflammation and other problems in the surrounding tissue. And typically, injured cells can be described as cells that are undergoing necrosis. Some of the characteristics of necrosis are cell swelling, broken membranes, a depletion of ATP within the cell, inflammation of surrounding tissue, and the DNA is randomly fragmented. This is an image of a cell undergoing some form of necrosis, and you can see that the membrane is not intact and the cellular contents are leaching out into the surrounding area, which will cause major problems for the organism. So, getting back to apoptosis as a systematically programmed series of events, apoptosis can be characterized by cellular condensation, so the cell starts to shrink. The membranes remain intact during this process. It's an energy-dependent process driven by ATP. The cell that remains can be phagocytosed by the surrounding tissue. The DNA of apoptotic cells is fragmented in a non-random way, and apoptosis can occur in individual cells and not affect the surrounding cells. So, one of the defined morphological changes that occurs during apoptosis is chromatin condensation and nuclear fragmentation. So effectively, it's been noted that the DNA of apoptotic cells breaks down into regular sized fragments, and this is referred to as DNA laddering. So this little animation here shows the chromatin or DNA of cells being broken up into a series of fragments. Now, these fragments may look fairly random, but when you run them on an agarose gel, you see a regular pattern of banding. So the size of this band is a multiple of this, and the size of this is a multiple of that. So the DNA is being broken up at regular intervals to produce this laddering, as opposed to just a smear of random fragments that you might see on a gel. Another defined morphological change that occurs during apoptosis is the flipping of the phosphatidylserine residues from the inner leaflet of the membrane to the outer leaflet of the membrane. So effectively, phosphatidylserine residues become exposed on the plasma membrane. So effectively, a cell that's undergoing apoptosis shrinks, it condenses, the membrane blebs to form these small bubble-like structures, the chromatin of the cells becomes fragmented, and we haven't talked about this but we'll look at it soon, is that the mitochondria of apoptotic cells breaks down and cytochrome C is released into the cytosol, and also the phosphatidylserine, which is normally hidden in the inner membrane, is exposed on the surface. And we'll talk about two types of apoptosis. We'll talk about apoptosis that is triggered by an intrinsic pathway, meaning a pathway inside the cell, and this pathway typically involves the mitochondria. And we'll talk about apoptosis that is triggered by external factors. So this is an extrinsic pathway. So there are receptors on the surface of the cell which trigger apoptosis. In both cases, apoptosis is mediated by proteins that are referred to as caspases. So these caspases mediate the cell shrinkage, the DNA fragmentation, and other changes that have been described. Caspases are a class of proteases, so they're proteins that cleave other proteins. Now, they're called cysteine proteases because they have a cysteine residue in the catalytic site, and they cleave other proteins adjacent to aspartate residues. So the term caspase comes from the fact that they're cysteine aspartate proteases. Now there's a family of caspases and they have common features. These common features are a cysteine residue in the active site, so there's a conserved motif sequence, and caspases cleave a peptide bond in the target protein C-terminal to an aspartate residue, so on the C-terminal side of an aspartate residue. We'll be looking at caspases that are referred to as group 2 caspases, and these are the ones that are particularly involved in apoptosis. Now, caspases are activated by proteolytic cleavage, so this tells you that caspases are initially synthesized in an inactive form, and through proteolytic cleavage of these caspases they become active. Now, the first events that triggers the activation of caspases is an autocatalytic event, so they can cleave themselves to become active. Okay, so that's not surprising. If you think about it, caspases can cleave other proteins, caspases can also cleave themselves, and as they cleave themselves, they can then assemble into a more efficient structure which is able to cleave other proteins. Caspases can be classified as either initiator caspases or as effector caspases. Now, when apoptosis occurs, you get a cascade of caspase activity. This is started through the initiator caspases, which auto-cleave themselves, and members of the initiator caspases are caspase 2 and caspase 9. These caspases can also cleave and activate downstream caspases, such as these effector caspases, and these are referred to as caspases 3, caspase 6, and caspase 7. So as I've already mentioned, caspases are synthesized inactive, and these are referred to as procaspases. Procaspases need to be cleaved to become active, and this first cleavage event is an autocatalytic event. And once caspases are active, they can then cleave other caspases. The caspases that activate first are referred to as initiator caspases, and these are able to cleave and activate effector caspases. And through this activation process, you get an amplification and a cascade of caspase activity. So this is showing the primary sequence in a very general sense of the caspases, and we have a pro domain, so in the pro caspase this domain's intact. Activation through cleavage, there's a cleavage site here and a cleavage site here, so the pro domain is removed, giving rise to two subunits, a small subunit and a large subunit. Now these subunits can come together to form a tetramer of two small subunits and two large subunits, and this complex constitutes an active caspase. So what are some of the targets of caspases to give rise to some of the morphological changes that have been observed? One of these targets is an inhibitor protein called ICAD. And ICAD can be cleaved by caspases, and therefore the inhibitor is removed. Now, ICAD is an inhibitor of a caspase activated deoxyribonuclease. So the CAD protein is a deoxyribonuclease. This means it's an enzyme activity that can cleave DNA. So this activity is inhibited by ICAD, an inhibitor of a deoxyribonuclease. Caspases cleave the inhibitor, release the deoxyribonuclease, leading to DNA fragmentation. So there's a very simple animation here showing that caspase 3, once it's activated, can cleave the inhibitor of the deoxyribonuclease. And once the inhibitor of this deoxyribonuclease is cleaved, the deoxyribonuclease can cleave DNA. Okay? So one of the morphological changes seen during apoptosis is regular fragmentation of chromatin. And this is instigated by a deoxyribonuclease which is activated by a caspase. So caspases are involved in apoptosis. There are two main mechanisms to trigger apoptosis. One of these is triggered by internal signals, and one of these is triggered by external signals. The important point is that both of these mechanisms of triggering apoptosis use caspases to mediate their effect. So the intrinsic pathway to trigger apoptosis involves the mitochondria. Typically, the mitochondria is thought of as being an organelle whose major function is to generate ATP as an energy source for the cell, but it also has an important role to trigger apoptosis. The mitochondria is very sensitive to DNA damage and other internal factors that can trigger apoptosis. These factors cause the mitochondrial membrane to become more permeable, and once the membrane's permeabilized, it allows the release of proteins from the mitochondria. Now, one of the proteins that is released is cytochrome C, and the permeabilization of the membrane is governed by a family of proteins referred to as the BCL2 family of proteins. So the BCL2 family of proteins affect the permeability of the mitochondrial membrane, and once the membrane is made permeable, you can then release factors such as cytochrome C into the cytosol as a trigger for intrinsic apoptosis. So what is cytochrome C? Cytochrome C is a protein that's normally involved in the electron transport chain. So it's normally involved in shuttling electrons through the electron transport chain. Cytochrome C is not an integral membrane protein, but it's loosely associated with the membrane. And cytochrome C resides in the outer face of the inner mitochondrial membrane, so that it can be released. So in a healthy mitochondria, the outer membrane contains the BCL2 protein and the Bax protein on their surface. And the BCL2 protein is bound to APAF1, which is an apoptotic protease activating factor. So BCL2 is not just the name of an individual protein, but it also refers to a family of proteins. And within this family of proteins are factors that promote apoptosis and factors that inhibit apoptosis. So one of the main members of this family of proteins that triggers apoptosis is the Bax protein. And one of the main members of this family that is anti-apoptotic is the BCL2 protein. So it's the balance between the BCL2 protein and the Bax protein that govern the permeability of the membrane. So effectively you have anti-apoptotic factors and pro-apoptotic factors, and it's the balance between these factors that govern whether the mitochondria will trigger apoptosis or not. So the release of cytochrome C from the mitochondria has two effects. Firstly, it triggers apoptosis, and secondly, the electron transport chain can no longer function, so ATP production is compromised. So apoptosis is triggered by internal factors. So internal damage to the cell triggers BCL2 to break away from APAF. It also triggers the Bax protein to move to the outer mitochondrial membrane, making it permeable. This triggers the release of cytochrome C to leak out of the mitochondria into the cytosol, and the combined effect of cytochrome C and the APAF then cause caspase 9, which is one of the initiator caspases, to become active. So the initiator caspase then causes a cascade of caspase activity and therefore apoptosis. There's a diagram from the textbook here showing intrinsic apoptosis. So there's some internal effect in the cell, some internal damage or stress that occurs in the cell, and this causes a pro-apoptotic protein such as Bax to move to the mitochondria membrane. This causes a change in permeability of the membrane, allowing cytochrome C to be released, and also the APAF protein is released from various factors, and the combination of the cytochrome C and the APAF allow this apoptosome to form, whereby these two factors and procaspase 9 aggregate, causing the autoproteolytic activation of the procaspase, and then this leads to the activation of apoptosis and other caspases. Apoptosis can also be triggered by extrinsic factors. This pathway involves a plasma-based receptor. These receptors are referred to as death receptors, and these receptors effectively bind an external ligand and trigger a transduction leading to the activation of apoptosis. There's a schematic in the textbook of showing the primary sequence of one of these death receptors, and as you can see here, they have a transmembrane domain, they have a ligand binding domain, and they have a death domain. So here's a schematic from the textbook showing one of these death receptors on the plasma membrane. Now the series of events is that a ligand binds to these death receptors. This causes a trimerization of these death receptors. So a collection of 3 of these receptors come together. The aggregation of these death receptors also brings together these adapter proteins, and the adapter proteins are able to bring together the caspases, and this leads to the activation of the initiator caspases. So through ligand binding, the procaspases are activated, and then these initiator caspases can then cleave and activate effector caspases, giving rise to a caspase cascade. The adapter proteins here, an example would be the FAD proteins, and these also contain other domains referred to as death effector domains. And it's these death effector domains on the adapter which can bind and bring together the caspases, leading to the initiator caspase activation. And bringing both the intrinsic and extrinsic pathway together, there's a diagram here from Hancock showing the extrinsic pathway leading to the activation of initiator caspases, and it's showing the intrinsic pathway with the mitochondria leading to the activation of caspases and the apoptosome. And effectively, these two complexes lead to the activation of effector caspases and then the morphological changes that we observe during apoptosis. Alright, well, I hope this summary was useful to you. Thanks.